Well, I'm glad we all got in here in this terrible storm. I want to welcome you to this 2006 series of our Biomedical Research Caucus. You know we have always open to any suggestions you might have about topics. This year we've already discussed genetic flu, cancer, Alzheimer's, and we should have a very interesting topic today. I just want to mention, just for one digression, one aside. In all the 35 years I've been here, this really is the worst year for the NIH. I've never seen it so grim. Really, it began when, and it's a bipartisan effort. After 5 years of doubling, when we were able to double the NIH and get up to $28 billion, in which our 4 co-chairs played a major role: Sherry Bullock, Rush Holt, Lois Capps, and also our new one, Mike Castle from Delaware. They played a major role in the doubling, and we're delighted. But this year, funding has just slowed down. We're waiting apparently till the fall, maybe even after the election. We just want you to keep in mind in your offices, the time will come up when the labor HHS bill will come up between the House and the Senate. And at that time, there may be some other funds around. Actually, any advocates that work for the NIH and other people realize very quickly that the NIH has widespread favor throughout the Congress. People look at it as the crown jewel of federal efforts in science, innovation. So we need desperately this fall to have your help, and I'm sure we will through your offices. Today, we will learn about recent scientific advances that have identified common variants in human DNA sequences, breakthroughs that will lead to individualized patient care in the near future. Also, how genetic information will be used to find risk factors for several common diseases. And we are really privileged to have with us a world-renowned scientist in the field of genomics. Dr. Kelly Frazer joined Perlegen Sciences in January 2000 with a strong background in the field of genomics. She received her PhD from the Biomedical Sciences program at the University of California, San Francisco, and then was a postdoctoral fellow in the Genome Science Department of Lawrence Berkeley National Laboratory in California. From 1997 to 2000, Dr. Frazer held a research scientist position at that laboratory. She's currently leading a genome-wide SNP discovery project in the mouse genome, and in collaboration with scientists at the Whitehead Institute, is characterizing the segmental structure of variation in inbred strains. We really look forward to her briefing. Dr. Frazer.
Thank you very much for having me come present today. I appreciate the opportunity. My talk is actually structured into 3 separate sections. The first part of my talk, I'm going to discuss how we've recently established the relationship between common SNPs in different human populations. This is the efforts of the HapMap project. This first part I'm going to spend about 10 minutes describing, and it's going to be relatively technical compared to the rest of the talk. So it's going to be about 10 minutes of pretty technical science, but I felt that I needed to describe the science in the terminology that we typically use. And because this has been such exciting achievements that have been made, I wanted to share it with you. And then I'm going to spend the next 10 minutes describing how we're using that information that we've uncovered, this structural relationship of common polymorphisms in the human genome, to identify genetic risk factors for human diseases and individual variation in drug response. And again, I'll spend about 10 minutes discussing this. And then in the last 5 minutes, I'm going to discuss possible means for applying the knowledge of genetic risk factors in order to improve human health care. So human beings have highly similar genomic sequences to one another. At the DNA level, are ATCG, those string of nucleotides that we have. When you look at the DNA sequence of one individual, it's highly similar compared to the DNA sequence of another individual. However, when we look at people, there's vast phenotypic, physical trait, complex trait differences between people. People have different skin color, they have different heights, they're different weights, they have different propensities to develop different types of diseases. And these different complex traits arise from the polymorphisms, the sequence differences, the base pair of differences that are present in all of us. So single nucleotide polymorphisms, which are commonly referred to as SNPs, this is the most common type of variant in the human genome, the most common type of difference that exists between different copies of the human genome in different individuals. And what a single nucleotide polymorphism is, it's a position in the human genome that's biallelic. So if we have two copies of the human genome, we have copy number 1, DNA 1. It's again a string of nucleotides which we designate as ATGC, and another copy from a different individual. What you'll notice is that by and large, most of the nucleotides are the same, except at some position they're variant. These are called single nucleotide polymorphisms, variants of the single base pair. Now, if we look at 100 different copies of the human genome, and the T allele, or the T base pair, is found in 15 of those, and the A is found in 85. We refer to this as allele frequency. So we would say that the SNP allele frequency is 15% and the T allele frequency is 85%. Another jargon that I'm going to use a lot is the word genotype. When we genotype a SNP, what we're doing is we're looking in a given individual and we're determining what that individual has at that position. Do they have an A? Do they have a T? Are they a TT, AA, or AT? Now, single nucleotide polymorphisms, there are approximately 8 million common types of single nucleotide polymorphisms in the human genome. Interestingly, these single nucleotide polymorphisms have arisen once in human history, and thus individuals that have the same base pair difference share that base pair difference because in the distant past they've had a common ancestor. So irregardless of our ethnicity, where our ancestors come from, if I have an A at a particular base pair position and you have that same A allele, we have that because at some distant point in our history we have had a common ancestor. Within a given individual, the majority of the SNPs are common, meaning most of the differences that are most of the changes that are in my particular genome are shared in common with a vast number of other people. Now, in early 2001, there were 2 human genome record sequences published. One was the effort of the private sector, and one was the effort of the NIH, the public sector. And there was a lot of celebration. This was a huge achievement. But in order to use this sequence data to improve healthcare based on biological differences between people, we had to uncover what the common variants in the SNPs were. These had to be discovered and characterized. So all we had in 2001 was the reference sequences. We knew 2 copies of the human genome, and that's it. There was no way we could use this information to improve public health care because we didn't know how different people varied in those positions. So the goal of the HapMap project, as well as Perlegen Sciences, initially was to discover common human polymorphisms, common human SNPs. The HapMap, which was a public effort, discovered a few million SNPs. Perlegen Sciences, independently working, discovered a few million SNPs. We used different procedures to do this. And what you can see here is this is the number of SNPs in millions that have been deposited in the database over time. And in late 2003 here, you can see that there were a few million SNPs that were currently well verified and known in the public database. And this was a sufficient number of SNPs to allow us to start characterizing how SNPs vary in different humans. So to do this, we wanted to establish the relationship between common SNPs in different human populations. So the experimental design was to genotype 270 individuals. Now these 270 individuals were collected as the NIH-sponsored HapMap project. That was phase 1. Perlegen was a strong participant in the phase 2 part of this project and actually performed the genotyping. Again, genotyping is when you look at a particular SNP and determine in a particular individual whether that individual has an A or T or AATT at that position. And we looked at 270 individuals of diverse ethnic backgrounds, 90 Yoruba from Nigeria, 90 European descents from Utah, 45 Hmong Chinese, 45 Japanese, and we genotyped them for 4 million SNPs. And we're in the process, and we have significantly done a lot of analysis, but we're still in the process of analyzing this data set to determine the correlational structure of these SNPs. One more technical thing to bring up, and that's this concept of linkage disequilibrium. Now remember, SNPs, a SNP is a biallelic site, meaning that it can have one of two nucleotides at that. So if you have 2 biallelic sites, then you can have 4 combinations. So if we have 2 SNPs, we can have an AA combination, we can have an AC combination, we can have a GA combination, or we can have a GC combination. Now if you look at 100 copies of the human genome and you only observe 2 of these combinations, these SNPs are in complete correlated with one another. They're in what we call complete LD, meaning that if I determine that an individual has an A at SNP1 position, I know automatically that they have an A at SNP2 position. Whereas if I look at 100 people and these 4 combinations are present in equal amounts, they're completely unlinked, meaning that if I type this person for SNP A, I have no idea what the position at SNP2 is. So this is the LD bin structure of a 190 kb interval containing the CFTR gene. This is the gene that's mutated in cystic fibrosis disease. And I'm showing it for 3 populations: African Americans, Caucasians, and Chinese. But I'm going to focus more on the Caucasians to simplify this. So this is the position of the actual gene in the genome. These triangles are indicating positions of the SNPs. And then what you look here is you see these bars represent the positions of the SNPs. If you come up, you find a triangle. What these bars here, what the horizontal bars are indicating, are SNPs that are in LD with one another. So there's 7 LD bins here. Again, an LD bin is a group of SNPs that are all highly correlated with one another, meaning that if you genotype one of those SNPs, you gain information about the entire group. You know what the combinations are. So by looking at this, we had to empirically determine this. There was no way that we could determine this except looking at these individuals and analyzing the data. And that's because you can see that this SNP is actually correlated with a SNP that's quite far away from it, like over 100 kb. That's quite a big distance in the human genome. However, with a SNP that's right adjacent to it, you can see that they're physically on this line adjacent to one another, they're uncorrelated. So there was no way that we could look at the sequence and determine these types of correlations. We actually had to empirically determine it. And what we've done is we've determined that the majority of common polymorphisms in humans are in these types of LD bin structures. The Yoruba from Africa have fewer of their SNPs in LD bins. It's known that Africans have a higher diversity in their genome than Caucasians or Asians. Caucasians and Asians have about 88% of their variants in these types of LD bin structures. And what this has allowed us to do is select sets of tag SNPs, which we can genotype. Remember, there's 8 million SNPs in the human genome, and we can genotype just a subset of those SNPs, 325,000 of those SNPs, and gain information in a particular individual about a large fraction of the variation that individual has in their genome. This is a huge technological advancement. So in summary of this first section here, the reference human genome copies were determined in early 2001. Since that point in time, we've discovered a significant fraction of all common SNPs in the human genome and have characterized them. The majority of these common SNPs are correlated with one another and are in LD bins and correlated with other SNPs. This has allowed us to select a subset of SNPs that we can then go in and investigate the genome of any given individual in a technically feasible manner and uncover the larger and determine what the genetic variation in that particular individual is across their entire genome. Again, a major achievement. So now that we have achieved this, we're able to start moving forward, and we're able to start identifying genetic risk factors for common human diseases and variation in drug response using case-control genetic association studies. And what I'm going to do is I'm going to focus on 3 particular case-control genetic association studies, just to give you a flavor of what the field is doing. The first one will be an olanzapine treatment emergent weight gain. The second one is going to be focused on adult macular degeneracy. And the third one is going to be on type 2 diabetes mellitus. So what is a case-control genetic association study? This is where we take cases, individuals that have a particular complex trait, a particular disease, a particular adverse reaction to a drug, and we take controls, individuals that do not have that complex trait or adverse reaction. And preferably we have about 1,000 of each of these, and we genotype the DNA of these individuals looking at these polymorphisms. And then what we do is we find associations with the SNP alleles with a given with the given complex trait that's being examined. And then what we want to do is we want to independently replicate the findings using other populations, and this is to assure that the initial associations that we have found are good, they're verified. And then secondly, we also want to validate findings using biological evidence and other types of things that this particular gene that's been associated actually has a reasonable functional role in the trait, in the phenotype trait. What exactly are we looking for when we're saying that we're trying to identify SNP alleles associated with a complex trait? Well, this over here on our left is our cases. They have an undesirable complex trait, and hence they have unhappy orange faces. Our controls do not have that undesirable trait, and hence they know they don't have that, and they're quite pleased about it. So our SNP allele here is designated as a green red. One SNP allele for this position is red, the other is green. And what we're looking for here is for the frequency of the SNP allele. And we see in cases that it's present at a 40-60%, so 40% have green and 60% have red, whereas the controls, 50% have green and 50% have red. And this is exactly what we're looking for. If a gene or a SNP allele is associated with a trait, the cases will have a different SNP allele frequency than the controls. So when we're genotyping these populations, that's what we're looking for, SNP allele frequency differences between the cases and controls. Okay, so to discuss our first genetic association study, olanzapine treatment emergent weight gain. Olanzapine is an antipsychotic drug which is used to treat schizophrenia. It has a reasonable efficacy, but it has a really bad side effect. After 6 to 8 weeks of treatment, some individuals are fine. Other individuals, their schizophrenia has been treated. However, they've had tremendous weight gain. And we're talking about weight gain of up to 50, 60, 70 pounds in some cases over the course of 6 to 8 weeks. Tremendous health hazards. I've had doctors tell me that they won't use this particular drug just because of the small fraction of individuals that have this huge weight gain. So this particular study was performed by Perlegen Sciences with Lilly. Lilly is the olanzapine is a drug that they produce. And what we did is we genotyped 255 cases, individuals that had tremendous weight gain, a mean body mass index change of plus 7.8. Again, a body mass index is the weight in kilograms divided by height in meters squared, and 258 controls, where they had an actual minus mean body mass index change. We took the DNA of these individuals, genotyped them, and found an association in the SNP in the HD1 gene. The particular product of this gene is located in cilia with a concentration in the basal body, and cilia are these protrusions that stick out into lumina, into areas, vacuoles, spaces in the small intestine, kidneys. They're found in various organs of the body. And it's believed that mutations in genes like PKHD1 and others maybe result in failure of the cilia to correctly be able to sense the environment that's going on. This finding, as well as there's been other findings in the field of obesity recently, that's suggesting that cilia may provide targets for obesity. So previously, a few years ago, there was no idea that cilia would play this role in obesity. Our collaborators at Lilly knew that Greg Germino at Johns Hopkins University was making loss of function mutation of this particular gene that had been associated, the PKHD1 gene, in mice. And working with Greg, they have shown that mice with this particular mutation have a marked accumulation of visceral fat, again indicating that that this particular gene, or strengthening the functional association of this particular gene with olanzapine emergent weight gain. And then finally, in an independent obesity sample using trios, so this is a family structure here, parents of normal weight and a programmed child with extreme obesity, a body mass index greater than or equal to 40. Looking at obesity trios of this nature, we again found an association of this gene with the obesity phenotype. The frequency of this particular SNP allele predisposing to this weight gain was 52%. So this is a quite frequent SNP allele in the general population. It's present in many of us. Having one copy of this allele gives an individual 1.3-fold relative risk of developing obesity. 2 copies of the allele a 1.6-fold relative obesity. And the fraction of obesity attributable to the SNP, meaning that if this particular risk allele was taken out of the general population, approximately 24% of the cases of obesity would no longer be present. Moving on to the second genetic association study based on age-related macular degeneracy. The macula is located in the center of the retina, and what it does is it converts light into electrical impulses, which it sends to the brain, allowing vision to occur. In age-related macular degeneracy, the macula degenerates. This leads to a loss of central vision, and it's the leading cause of blindness in individuals over 60. Age-related macular degeneracy is a complex trait. It has this age component of occurring in late in life. There's environmental as well as hereditary risk factors that are involved in this. There have been many advancements, particularly in this particular disease, in the last year, and the genetic understanding of this disease is really, really increasing. This is one study by Klein et al. In Science that was published last year. There were a few other papers that were published simultaneously, but in this particular study, they genotyped 96 cases and 50 controls. So this is normal vision. This is what a normal person would see if they were looking at black and white. And this is the view by a person with age-related macular degeneracy. Again, the central vision is completely destroyed. By doing this particular association study, they found an association with a SNP in the complement factor H gene. This was a new surprise. Again, a new mechanism was uncovered that was underlying this particular disease. The complement cascade is a molecular pathway which it's the body's first line of defense against bacteria. But if it's overreactive, it's known that it can actually destroy tissue, and that's exactly what's going on here. It's been known for a long time that there's these yellow deposits which are called drusen. This is the retina. You can see the yellow deposits here. It's been known for a long time that these yellow deposits are correlated with AMD, age-related macular degeneracy, and now after the finding that this complement factor H gene was associated with this disease, they've looked more closely at these drusen, and they found that these abnormal deposits contain complement proteins. So the frequency of the SNP predisposing to AMD is 39%. Again, this is a very frequent gene present in many of us in this room. The frequency in cases is 61%, in controls is 34%. Again, we're looking for SNP allele frequency differences between people who have the disease and people who don't. The relative risk with one copy of the allele is 2.8. 2 copies give you a 6.3%, 6.3-fold relative risk. The percent attributed to the SNP allele is 62%. If this particular SNP allele, risk allele, was taken out of the population, 62% cases of AMD would automatically disappear. Now finally, the third thing I'm going to talk about is genetic association study for type 2 diabetes mellitus. The pancreas, which is situated in the body cavity here by the stomach, these are the kidneys, this is the pancreas, produces insulin. And the role of insulin is to lower the blood sugar level. Type 2 diabetes is the most common type of diabetes in individuals over 40. And what happens is peripheral insulin resistance occurs. This is where the body in people who have type 2 diabetes can no longer respond appropriately to insulin. And there is sometimes insulin secretory defects, but this is secondary and varying problems. Now, again, type 2 diabetes is a complex disease. There's environmental as well as hereditary risks involved. In this particular study, there were 226 cases and 210 controls that were genotyped. This is Grant et al., which was published earlier this year. This is a study that was done by Decode. And what this is supposed to indicate here is a normal cell in our controls where there are insulin indicated by the yellow, insulin receptors indicated by this orange hook, glucose. The insulin receptors, insulin are working properly, allowing the cell to absorb glucose. In the type 2 diabetic cell, again, all the components are there, the insulin receptors are there, the insulin's there, the glucose is there, but it's not working appropriately, so the glucose is not being absorbed correctly. Genotyping 226 cases, people with type 2 diabetes, and 210 controls resulted in the identification of an association with a SNP in with a transcription factor. This is known as TCF7L2. So this particular transcription factor is hypothesized to be involved in blood glucose homeostasis. Just to remind you what a transcription factor is, this orange loop right here is a double helix DNA. Transcription factors are proteins that interact with the DNA directly in order to transcribe it, to make messenger RNA. The frequency of the SNP predisposing to type 2 diabetes is 28% in the general population. Again, quite frequent, common in many of us, present in many of us here in this room. The cases have a 39% frequency, the controls at 25%. The risk with one copy of the allele is 1.6. 2 copies increase your relative risk 3.3. And if this risk allele was removed from the population, 28% of the type 2 diabetic cases would be resolved. So the conclusions here are that genetic association studies identify risk factors associated with common disease and variation in drug response. We're having success in this field. These genetic risk factors are SNPs. They're the variants. So SNP alleles that are more frequent in cases than the controls. However, it's really important to remember that not all cases have the associated SNP allele, and not all controls lack the associated SNP allele. Therefore, any single genetic risk factor is responsible for only a small contribution to the complex trait. Most of these complex traits, therefore, are caused by 20 or more genetic risk factors which are distributed across the genome, and we have to get a good handle and understanding of the majority of these genetic risk factors as well as the environmental risk factors that an individual is subjected to in order to use them in aggregate in a clinically useful manner. Now, for the last 5 minutes, I just want to talk about how we're going to attempt to apply the knowledge of genetic risk factors in order to improve human health care. There are many different scenarios that one could come up with. This is just one that I believe would be a good manner to go about it in. And that we would require 2 things. One is implementation of policies preventing genetic discrimination. And secondly, it would require establishing a regulatory system for monitoring outcomes of prescribed drugs and medical devices. And this regulatory system would have to be integrated into the healthcare practice,
and it would require public and private sector partnerships. So it would have to really encourage the fostering of those types of partnerships. Why do we have to enact laws to prevent discrimination, genetic discrimination? And that's because many Americans fear that genetic information is going to be used against them. That it's going to be used against them to deny, limit, or cancel their health insurance, and then it may be used against them in discrimination in the workplace environment. This fear of discrimination is resulting in individuals deciding actively not to participate in NIH-funded or NIH research or clinical trials that involve genetic testing. This fear of discrimination is also resulting in patients refusing genetic tests from doctors, which they want to administer in order to help diagnose and treat their particular disease that they feel that the individual may have. Until Americans are no longer fearful of the manner in which genetic information will be used, we cannot push forward the use of genetic information in order to improve human health care. Secondly, we need to put into place a regulatory system for measuring clinical outcomes of prescribed drugs and medical devices. Carefully tracking risks and efficacy in hundreds of thousands or millions of patients that go on to a new drug or new therapy really would address the power issues that currently limit findings in moderate-sized trials. Currently, the information that's gotten out of moderate-sized trials, as we've seen in recent years, the exact efficacy of a drug is not known. The exact side effects of that drug is not known. Are there rare side effects that are going to come up? And we need to monitor, as we put populations on these medical devices, what is happening to the individuals going on them. This would require collecting DNA samples, data on the environmental factors, as well as clinical measurements. So this would most be beneficial if this regulatory system was actually integrated into the healthcare practice. So in theory, we separate medical research from medical practice in order to prevent unnecessary risk to the patient. We don't want to put patients at risk by performing research on them. But we are putting them at risk by putting them on these new drugs that are not completely understood, all the side effects, efficacy issues, etc. People are at risk all the time when they go on these new things. And by having a monitoring system in place, what types of risk do we add? Well, we do add risk. If people are being monitored, we are adding the risk that there are privacy issues, and we're adding risk that there may be genetic discrimination. But we will need to take care of these types of things. In many cases, the benefit of a drug or a medical technology will be limited to a specific population, while other patients will have higher than average risk for adverse outcomes. And by monitoring how a drug, medical device is working with genetics, environments, and other aspects, we'll be able to subset populations better. This is going to result in increased likelihood of improved health outcomes because we're only going to put people on these medical devices, drugs that are going to benefit, and it's going to also result in reduced health care because people who will not respond, have poor outcomes or adverse effects, will not go on these medical technology or drugs, and will not have the resulting complications, which will result in further cost. So this regulatory system is really going to require a lot of public and private sector partnerships. It's going to require a lot of interactions of entities that are currently acting independent from one another, all with the general goal of increasing human health care, but doing it in a rather independent way. A system of this manner is really going to have qualified scientific oversight. There's going to have to be appropriate clinical, scientific, technical expertise in many different facets with many different people. Data analysis and dissemination of that analysis, how's that going to occur? So it's really going to require the NIH funding for medical purposes, biotech industry, pharmaceutical companies, healthcare insurers, as well as healthcare providers, working more closely together. There has been some movements that have been made in this There's this GAIN, this is a Genetic Association Initiative Network, which was announced earlier this year, which is a unique public-private medical research partnership between the pharmaceutical company Pfizer, the NIH, and Perlegen Sciences. We're looking to see, can we uncover additional genetic risk factors for common human diseases? And then Dr. Mark McClellan at the CMS, the Center for Medicare and Medicare Services, has put forth a draft document for coverage with evidence discovery, saying that medical intervention that has some evidence that it's going to be beneficial can be covered and be monitored to determine whether that evidence was strong enough to continue its application in the medical field. This will be bringing together biotech industry, pharmaceutical companies, healthcare insurers, the CMS itself, as well as healthcare providers. So in summary, we've had tremendous gains in this field through the NIH-funded HapMap project. A significant fraction of all common variants in the human genome have been discovered and characterized. This is tremendous scientific achievement. Genetic association studies are currently ongoing right now, and they are identifying risk factors associated with common disease and variation in drug response. However, in order to apply the knowledge of these genetic risk factors to improve human health, various structural changes have to occur, including implementing genetic non-discrimination laws and developing regulatory systems for measuring clinical outcomes for new treatments and drugs. Thank you very much.
Thank you, Dr. Frazer. And now we'll take questions for Dr. Frazer. And we have a mic floating around, so you can just wait a second for a question. But let me ask you the first question.
Yes.
How will this change your going to your general physician for an examination and being, if you feel ill about something, how will it change all that?
I think that it will not change the initial going to the general physician. Right now you go to a general physician, you describe what your symptoms are, you tell them you don't feel well, you have mid-gut pain, your mid-gut pain shifted to your lower right quadrant in the middle of the night. The doctor goes through a series of questions, and then based on that, he or she will order a series of tests for you to determine what your ailment is. And here we may be seeing an increase in not just the scans, the MRI scans that many of us have been undergoing recently, but also the requirements for, the genetic tests will also be included in that.
Thank you. Yes, sir.
Just real quick, I'm Brian Peterson with Congressman G. Bader's office. We're the chief sponsor of HR 1227. Appreciate all the things you said today about the importance of all this technology. I wanted to let everyone else in the room know that legislation currently has 214 co-sponsors, 99 Republicans, 115 Democrats. I think we're very close on this issue, and I think it's something that can happen in this Congress. So I brought a one-pager with me that I'll leave out front of you. Who wants to grab a copy, feel free to talk to me afterwards if you have any questions. So.
Thank you. Thank you very much. Over here, please. Just 1 minute.
You didn't mention patent law. I'm wondering, how does current patent law either aid or hinder genetic research?
Can you explain to me a little bit?
Well, my understanding is that certain sequences can be patented.
Oh, patent law. Okay. So I am not aware of any particular case at this point in time where a patent has stopped genetic research. There are some perhaps abilities to bring in, like there's the Herceptin. There are some drugs where there are somatic mutations that occur on particular tumors, and individuals have patented the identification of that tumor with the efficacy of the drug, and those patents may be preventing the development of additional drugs, but that's no more, I don't think, any more delay than normal patenting on a particular efficacy of the drug.
Over here, please. Yes.
Hi, I'm Susan Fall from Congressional Research Service. when you mentioned the percentage of disease in your examples of maybe diabetes, if you got rid of that one allele difference, 28% would be of the disease would not happen. But then after that, you said that but these are multifactorial and there might be things at many places.
Yes.
Was there? Were you?
So that's a tricky thing, and as I was actually stating it, I was realizing that was probably causing confusion. So the attributable risk can actually add up to more than 100%, because smoking. If we eliminated smoking from the general population, right, a large fraction of diabetes, a large fraction of health risk would also disappear, right? They would vanish. But that doesn't mean that, the population attributable risk might be 60%. But that doesn't mean that that's, the only thing. There are multiple other factors. So many things many things interplay with one another, okay, contributing to the risk. And if you take out any one of those components, then the overall risk would drop by 24%. But that doesn't mean that it's actually playing 24% of the general risk. Does that make sense at all?
A little bit.
Yeah?
Thanks.
Well, let me ask you another question, Dr.
Yes.
Frazer. You said that this would reduce costs, healthcare costs. How would it do it specifically and concretely?
How would it
How
reduce
would it?
healthcare
Yeah.
costs? Well, if we could determine who would benefit from drugs and who would not benefit from those drugs, and worse, who would have adverse reaction to those drugs, and these adverse reactions can result in, myocardial infarctions and very expensive health care down the way just by reacting to a drug poorly. We would then be able to, better put our money into giving people the drugs, the medical devices that would be beneficial for them.
Would physicians have to be specially trained to be able to use these kinds of procedures?
I don't think that a physician would have to be specially trained, no more than a physician has to know how to actually culture bacteria and to determine what antibiotic to use on that bacteria. There are people who are trained in that field, and they send these swabs that people send back and say, use this particular antibiotic, this particular strain is resistant to these. The physician doesn't have to know how to do that. They do have to know how to use that information.
Yes, sir.
Landon Huber with the Department of Health and Human Services. Also, I'm a student of biomedical engineering at the University of Utah, so I really enjoy this, especially the deep science part. From a business point of view, I'm trying to look at this not as a researcher, but as a business professional, let's say. When we start talking about personalized medication and looking at the costs of developing a drug, marketing a drug, and then getting that drug into the hands of the individual that's going to consume it, those costs can be to the hundreds of millions, even to the billion-dollar mark. When we're talking personalized medication and dealing with specific populations and specific disorders, how do we sell the business community? How do we incentivize the business community to want to put such extreme monies into doing something such as trying to get a specific drug for a specific population? Is that something federal money should be put behind? Is that something that should be a private sector initiative out of the goodness of their hearts? What is your perspective on that? I know you talked briefly at the end about partnerships. How should that be sold to the business community?
Well, I think that part of a regulatory system would be taking into account how are things going to be costed, right? So there's a lot of stakeholders in a regulatory system of this. There's the healthcare insurers, they have to pay money. There's the people that are going to be providing devices in healthcare, they're going to be making money. There's the the healthcare providers, which are going to have to be putting systems into place. So it's going to be a complicated thing that's actually going to involve, it's going to actually take a lot of thought of how to put it together. How to distribute those costs are one of those things. And it's not a clear path forward, but I think that by putting regulatory systems in place to monitor these things, doing share costing so that it's not all on one person, that everyone who's going to benefit from it will help provide the cost, will help answer some of these questions.
Yes, sir.
Hi, Scott Lovis with the Department of Health and Human Services as well. You mentioned public-private partnerships as sort of a way to encourage development of the medical technology in this area. Unfortunately, with the private sector, there's often a struggle with emerging personalized medicine and genetic information related to pharmacogenetics that they don't want to hear. Although they might like to know that their drug is effective in only a narrower slice of the population, or that certain adverse events could be avoided by doing genetic analysis of the potential patient taking this drug, it's not something that they really would like to hear. Again, the business case for the development of a particular drug is often based upon the population that would be taking it. And if you use genetic analysis and start to narrow down the case where the population is much smaller, the money case for moving the drug forward is often not really cost effective. And you end up in a situation where personalized medicine becomes more and more akin to an orphan drug issue because narrower slices of the population are afflicted or could benefit from a particular drug. How, as a researcher working in the private industry, would you think that there are ways for the public sector to try to go to the private sector and ensure that these technologies will still be developed, even though the business case may be against it?
Well, I mean, I think that there are ways that some of the examples that you put forth, I mean, some of the drugs that have been recalled from the market, if those manufacturers had been able to test to determine who would have adverse side effects, that the picture there would be a lot prettier right now. I mean, a lot of these companies are suffering from the onslaught of lawsuits. So I think that companies are opening up to it. In addition, we can think of it not only as narrowing the market for some drugs, but opening the market for others. So sometimes if you know that the first line of a particular drug that's used in the first line for a particular ailment is not going to be beneficial, what it does is it opens up room for that second line drug to come in. So there will be, for every drug that has a narrowing of the market, there will be other drugs that will be opening up that you'll be putting the individual on.
Kevin?
Where would this research be without NIH funding?
Well, the NIH is really a spearhead in all of this. So they spearheaded the HapMap project. The funding for that, they are really supporting a lot of the major scientists who are analyzing the human genome sequence, not only for this particular application, but for basic applications that will have medical relevance in the future. The funding of the NIH is very important for all of these.
Yes, sir. Just a second, sir. Have a mic.
Hi, Jim Campbell, House Budget Committee again. I'm wondering if you might look into the future for us. And what kind of breakthroughs will we see in 5 years, 10 years? We're going to have just gradual improvement over the next 50 years? Will cancer be cured in 2? Or how does it look like?
It's always hard because I think people always tend to.
Hard for people that know nothing about it.
Yeah. Well, people always tend to overestimate what they're going to accomplish in the next 2 years and really underestimate what they're going to accomplish in the next 10. So it's hard to know when, but I have a feeling that the genetic information, what it's not going to do is it's not going to be curing diseases. What it's going to do is there's going to be certain people that will be beneficial. So for like the AMD, the age-related macular degeneracy, there are numerous genetic factors that are involved in that. There are different responses to different types of treatments. What we can do is we're going to better align. So we'll take the genetic information, we'll take the environmental factors into account. We will be able to, more like just streaking that bacteria out and seeing what type of antibiotic do we use on this particular person to cure the strep throat, and what don't we want to use because the bacteria is already resistant to that. We'll be using a variety of information, genetic information will be one of them, in order to say this particular medication will work with the highest efficacy in this particular individual. They may have, they are likely to have, edema, swelling of the ankles, so tell them to be wary of that. If they, there's little chance that they'll have other types of side effects. So it will be more used in information to improve the health care. And depending on, many things depend on how society is going to react to this. If people are afraid that they're going to be discriminated against with genetic information, if there's not the systems in place to encourage and incorporate and support this, it's going to be longer. It will happen. I think that this genetic information is already beginning to be taken into account in the treatment of certain cancers, and it is coming. I would say in 10 years it'll be more routine than we imagine at this point.
Dr. Frazer, thank you very, very much for the interesting. Finally, we want to thank the Joint Steering Committee for Public Policy for sponsoring this luncheon. And I want to tell you that after your July 4th break, on July 19th, we're going to have a fascinating caucus on using the immune system to fight cancer. Thank you very much.